In this video, I'll be explaining the light-independent reactions of photosynthesis. This video follows the video on the light-dependent reactions of photosynthesis. If you have not watched that video, I suggest you do that first. Again, I recommend drawing along as I do so in order that you have a copy of the notes and to better understand the process yourself. I'm going to start by drawing the outer membrane of a chloroplast. All the reactions of photosynthesis happen inside a chloroplast. Chloroplast has got a double membrane, so I'm going to draw a second membrane on the inside of the first. In the previous video, we discussed the light-dependent reactions. And a reminder that those took place on the membranes of the thylakoids. Thylakoids occur in chloroplasts in stacks, with each stack being called a granum. So here's a thylakoid. And this whole thing, this is a granum. Many granum, the plural is a grana. So this is where the light-dependent reactions took place. The light-independent reactions take place in this liquid called the stroma. Think of it as being like the cytoplasm of the chloroplast. The series of reactions in the light-independent reactions are also called the Calvin cycle. This is a chain of reactions that happen in the stroma. Now, we're going to be drawing this as a cycle that rotates clockwise. But it's important to remember that all the components of this cycle, both the reactants, products, and enzymes that catalyze each of the steps, they're all moving around, mixing with one another in the stroma. I'm going to switch to a finer pen so that I can show you more detail as we talk about the different parts of the cycle. Now, because it's a cycle of reactions, we could kind of start anywhere. But I'm going to start with a reactant called ribulose bisphosphate. Ribulose bisphosphate is a 5-carbon molecule. And when I'm explaining the Calvin cycle, I think it's probably the easiest place to think about starting. Ribulose bisphosphate reacts with a molecule of carbon dioxide that has entered the chloroplast. Carbon dioxide can move in across the membranes of both the cell and the chloroplast because it is small and non-polar, so it isn't repelled by the fatty acid tails of these phospholipid bilayers. Carbon dioxide itself is very unreactive, so in order for this reaction to take place, we need an enzyme, and that enzyme is critical to know. It is called rubisco. Ribulose 1,5-bisphosphate carboxylase oxygenase. It's got a long name if you give it its full name, but you can just call it rubisco. This enzyme is the most abundant protein in the whole world. Why? For 2 reasons. One, it does a really critical job capturing carbon and fixing it in biological molecules from carbon dioxide. And 2, because in a way it's very bad at its job. So because it's bad at its, what I mean by that is it's very slow. You need a lot of rubisco to capture enough carbon dioxide. It also sometimes captures the wrong molecule. It sometimes captures oxygen, which causes all kinds of problems for the plant in a process called photorespiration. But we're not going to cover that here. The key thing you need to understand is rubisco binds the carbon dioxide to the ribulose bisphosphate. Now, that's a 5-carbon molecule binding to a 1-carbon one. That's going to make a 6-carbon molecule. But that 6-carbon molecule that results is extremely unstable and instantly splits into 2 3-carbon molecules. These 3-carbon molecules that are produced when this unstable 6-carbon molecule comes together, these are called glycerate 3-phosphate. They are a 3-carbon molecule. Glycerate 3-phosphate is often referred to by its abbreviation GP. Next, this molecule goes through a series of reactions. We'll draw some arrows down here to represent those reactions. In the first set of reactions, the GP is reduced. It's reduced by NADPH, which from the previous video you may remember is also called reduced NADP. This is a high energy electron carrier that was produced by the light dependent reactions. So the electrons from the light dependent reactions were transferred to this high energy electron carrier NADP to make reduced NADP, which in turn reduces the GP. This process will release NADP+, which in turn will go back to the light dependent reactions to pick up more electrons. The second thing that happens to the glycerate 3-phosphate is that it is phosphorylated. ATP phosphorylates the molecule, donating one of its phosphate groups to it. This results in a molecule of ADP, which will return to the light-dependent reactions. Remember, the light-dependent reactions were the source of ATP here. Learn more about the light-dependent reactions in the previous video. So these two products, NADPH and ATP, are providing high-energy electrons and phosphate groups, which are also providing energy for the glycerate 3-phosphate. All this transforms the glycerate 3-phosphate into a different 3-carbon molecule. This molecule is called Triose phosphate. Triose phosphate is extremely useful. Triose phosphate can be converted by the plant into lots of different useful organic molecules. A classic example of that, the one that we are most familiar with when we think of photosynthesis, is glucose. C6H12O6. This Triose phosphate, which is also, I should mention, sometimes just shortened to TP, Triose phosphate, you need two of those molecules because Triose phosphate is a 3-carbon molecule. So you're going to need 2 Triose phosphates for every glucose that's made. That's because glucose is a 6-carbon molecule. This process can produce other useful organic molecules, ultimately things like amino acids, fatty acids, glycerol, all kinds of different things that a plant will need in order to build its structure. It's also important to realize that this carbon coming in here and going round to Triose phosphate is the source of carbon for all living organisms, because even animals, right, they're going to get it this way by feeding on plants and gaining these organic molecules. Wait a second, this is called the Calvin cycle. It doesn't look much like a cycle right now. That's because we're missing one final step. I said that Triose phosphate can be used to make useful organic molecules, but not all of the Triose phosphate is. Most of the Triose phosphate will be converted back into ribulose bisphosphate. This process requires energy, and that energy is supplied by ATP, being broken down into ADP and PI. This phosphorylation of the Triose phosphate provides the energy needed to convert much of it back into ribulose bisphosphate. This ATP was also produced by the light-dependent reactions, and the ADP and PI will be able to return there in order to be regenerated into more ATP. So that's the basics of the Calvin cycle. One thing that can be confusing, though, is to explain how it is that some of the Triose phosphate comes down here, but the rest is needed to regenerate ribulose bisphosphate. Oh, I should mention, ribulose bisphosphate can be abbreviated to RuBP, like so. That can help if you're having to write it down lots of times. So, to review, ribulose bisphosphate, or RuBP, is bound to carbon dioxide by the enzyme rubisco. That forms an unstable 6-carbon molecule that rapidly breaks down into 2 molecules of glycerate 3-phosphate, also known as GP. These are 3-carbon molecules. The GP is then reduced by NADPH, gaining some high-energy electrons, and phosphorylated by ATP, becoming more reactive. The molecule is transformed into Triose phosphate, or TP. Some of the TP is used to make useful organic molecules such as glucose, which would require 2 molecules of TP. The rest is transformed back into RuBP by being phosphorylated by ATP, which provides it with energy. The confusion around the numbers and understanding this can be explained by thinking about the number of carbons in a bigger scenario. Remember, this cycle is happening lots of times, all at once. Let's imagine that we had to make 1 molecule of glucose. We would actually start with 6 molecules of ribulose bisphosphate, which has 5 carbons that I'm representing with little rings here. Carbon dioxide has 1 carbon. I'll represent that with 1 little ring here. When those join together, they make a 6-carbon molecule, but these 6 will join to 6 carbon dioxide molecules. 6 ribulose bisphosphate and 6 carbon dioxide will come together and then instantly split, so that we end up with 12 3-carbon GPs. Think about that for a second. We've got 30 carbons coming from here, 6 carbons coming from here. That makes 36, or 12 3-carbon molecules. These would in turn be converted into 12 3-carbon TPs. Now we said that we would need 2 of those to make our glucose. That would leave 10 behind, 10 of these 3 carbon TPs. Well, 10 times 3 is 30, which happens to be just the right number of carbons left to make our original RuBPs again. 6 times 5 is also 30. So 10 TPs can be converted back to 6 RuBPs, and the cycle could start again, gaining 6 carbon dioxides to produce 12 GPs, to produce 12 TPs, and that would release 1 glucose, 10 TPs that could be reverted back into 6 RuBPs. This is how we get the 6 carbon dioxides that are needed for photosynthesis in order to make 1 glucose molecule. I hope this has been helpful. Feel free to rewatch it again if you struggle to understand anything.